Hey everyone, my name is Sasha Ripley, and I'm going to be sharing with you a GED science class today about codominance and incomplete dominance. If you are interested in finding out more about my program, the Sasha Ripley LLC online GED course and virtual tutoring, you can find more information at my website, www.sasharipleyllc.com. So let's go ahead and get started. All right, so my program is my course, my science course. I also will be having social studies, language arts, and math courses as well. Is in the format of a Google Classroom, so I need to scroll down to get to my heredity and DNA section. So first, we're going to talk a little bit about review homozygous and heterozygous genotypes. All right, so the prefix homo means the same and hetero means different. A homozygous genotype has two identical alleles. In this case, we're talking about uppercase B and lowercase b. So our two homozygous genotypes are going to be two uppercase b's or two lowercase b's. A heterozygous genotype has two different alleles. So in this case, it's going to be one of each, an uppercase B and a lowercase b. Now, this particular example is about complete dominance or simple dominance, where one allele is dominant over the other allele. So the dominant allele will be expressed over the recessive allele. All right, so homozygous dominant genotype is going to be two uppercase B's. And in this case, my fish is going to be blue because blue color is dominant. And then I've got a heterozygous genotype, an uppercase B, a lowercase b. Still, my fish is blue because my uppercase B is the dominant allele and blue is dominant over yellow. Now I finally got a yellow fish over here because I've got two recessive alleles finally. When we're talking about complete dominance, that means that you do have to have two recessive alleles in order for the recessive phenotype to show up, in this case, yellow. So the homozygous recessive genotype is two little b's. All right, now let's get right into codominance and incomplete dominance. But just a quick example here. In my science course, in my GED science course, I just want to share with you that I do have notes included. So I basically created this course to make it the easiest, most efficient, fastest way for students to pass their GED exams. So I have 14 years of experience teaching GED classes, and I found that this is the fastest way to allow students to learn the information and to study. So these notes are there for you. If you purchase the course, you will find notes on all of the science concepts. And you can either print out the notes and study them that way, or you can just look at them on your computer at your own convenience. Okay, so going back here to codominance. All right, in codominance, both phenotypes are fully expressed, so the phenotype of the offspring is a combination of the parent phenotypes. So again, both phenotypes are dominant. If the bull is black and the cow is white, the calf is going to be black and white. You're going to have both of the parent phenotypes expressed in the offspring phenotypes. All right, so here I have some Punnett squares with some examples of codominance. This flower here, these are going to be flowers. This is a black flower with a genotype of BB, and this is a white flower with a genotype of WW. So now you are probably going to get a question or two about Punnett squares. Either you're going to have to fill out a Punnett square, or you are going to have to just analyze and read and answer some questions about a Punnett square that they give you on the GED exam. I found that the majority of my students over the last few years since the new GED exams have been created and gone out, Most of my students have gotten questions about Punnett squares. Now, we don't know which GED exam you're going to get. It's random, and it also depends on whether you take the in-person exam or the online exam. So I don't know what kind of question, whether it'll be like a straightforward complete dominance, whether it'll be codominance, incomplete dominance, maybe X-linked traits, possibly blood types. But I would expect that you would get some question about Punnett squares. So I'm careful to go through those in quite a bit of detail. All right, so this is how you read the Punnett square. You get 50% of your DNA from each parent. So you're going to get one allele from this parent and one allele from this parent. All right, so here, this is how we do it. We get this B from the first parent, this W from the second parent for BW, this B and this W for another BW in this offspring. Then we have this B here, this W for BW in this offspring, this B here, and this W for BW in this offspring. So out of my 4 possible offspring here, 100% of the offspring are going to have a genotype of BW and a phenotype of black and white, because again, in codominance, both of the phenotypes are expressed. So we're going to get the phenotype of black from this parent and the phenotype of white from this parent. Now again, genotypes and phenotypes. The genotype is the combination of alleles. So my genotype is BW. The phenotype is what's expressed or what you see. And in this case, the color of the flower would be the phenotype. That's what I can see. That's what is expressed. So I remember phenotype that phenotype starts with a P, picture starts with a P, and I can see a picture. So it's what I see or what's expressed. Now, it's not always something you can see. Sometimes it's some sort of like a disorder or a disease, but it's still expressed in the symptoms. Okay, so the possible phenotype percentages for the offspring of these parents are going to be 100% phenotypes of black and white. And the genotype percentages for the offspring are going to be 100% BW. All right, let's look at another Punnett square. I've got a red flower and a white flower. So I'm going to get an R allele, a W allele for RW, an R allele, a W allele for RW, an R allele, a W allele for RW, an R and a W for another RW. All right, so what are the possible genotype percentages for the offspring of parents with genotypes RR and WW? The answer is 100% genotypes RW. All of my offspring have RW genotypes. And what would be the percentage of phenotypes for the offspring of these two parents? That would be 100% red and white. Remember, phenotype is what you see. Okay, let's go on. One more diagram here for us to look at. Codominance again. So in this case, I have one parent that's black, one parent that's white. Again, you're going to get half of your genetic material from each parent. So this cow here can only donate a black allele. This cow here can only donate a white allele. So I know that all of my offspring are going to end up being black and white. All right, let's move on to incomplete dominance. All right, incomplete dominance. So again, remember that complete or that basic simple dominance, that is when the dominant allele completely masks the effect of the recessive allele. But we are now talking about incomplete dominance where neither phenotype is dominant. So the red and white phenotypes in these flowers combine to create a new phenotype of pink. All right, so let's look at this Punnett square. This flower is red. This flower is white. I get my R and my W, R and my W, R and my W, R and my W. So I have 100% probability of genotypes RW for my offspring. And though what happens here is instead of the flowers being either red or white or red and white, you come up with a completely new phenotype of pink. All right, so all of these offspring are going to have a phenotype of pink. Let's look at another Punnett square here. Now, this parent is RW and this parent is RW. So now I have two parents with heterozygous genotypes. So I get an R from here and R from here for RR, a W and an R for RW, an R and a W here, RW, a W and a W here for WW. So now I've got a little bit more genetic variability, right? A little bit more diversity in my offspring. I have 25% genotype probability of RR, 25% genotype probability of WW, and then I have 50% probability for my genotype of RW. If we're talking about phenotypes, it's 25% red, 25% white, and 50% pink. All right, one more Punnett square. This Punnett square has a parent with RW, heterozygous genotype, WW, homozygous genotype, R and a W, W and a W, R and a W, W and a W. All right, so now what are our genotype probabilities for these parents or the offspring of these parents? The genotype probabilities are 50% RW, 50% WW. So I've got phenotype percentages also of 50% pink and 50% white. Okay, one more diagram that we're going to look at here. Just another diagram with my flowers for incomplete dominance. So I've got my white flower parent here, my red flower parent here. All of my offspring are turning out to be pink phenotypes because I get a W from this parent and an R from this parent. Okay, so that covers incomplete dominance and codominance today. I hope that you have a much better understanding of these concepts. Just as a quick, some quick information for you, I just want to show you what if you were to be interested in purchasing the GED science course, you would receive access to the course for 4 years. And what I wanted to show you is what you would get in the course. So notice that we have video tutorials. So here for my heredity, I've got my codominance, incomplete dominance, Punnett squares, eye color Punnett squares, DNA and heredity, a pedigree chart, video tutorial, also lots of diagrams, blood types, blood type Punnett squares, X-linked recessive Punnett squares, pedigree charts, mitosis diagrams. Okay, so I also explain meiosis and mitosis in this particular module of my science GED course. And then also what you get besides all of the diagrams and the video tutorials, you also get worksheets. Okay, so I've got the worksheets in here for you to either print off and fill out, or you can just look at them on your computer and fill out the answers on a separate sheet of paper. But then what's very, very helpful is I also put the answer key right below it. Okay, so you can figure out if you got the questions correct or not. Okay, so again, thank you very much for joining me for this class about incomplete dominance and codominance. And keep in mind, even if you aren't getting your GED or HSCD, this course might be very helpful for someone who's in high school or middle school and possibly is just struggling with something. Say you're struggling to understand endothermic reactions and exothermic reactions. So all you have to do is go in here, find the video about endothermic and exothermic reactions and watch that. Or look at the notes about the endothermic and exothermic reactions, do some of the worksheets. Also, people who are homeschooled. Maybe you're a homeschool parent and you just cannot understand about balancing chemical equations. Well, if you purchase this course for your students, it's very easy, very organized. They will have no problem learning these difficult concepts. So think about it for other things, even besides GED and HSED. The course was created for that particular purpose, but it's definitely helpful for people, maybe even just trying to review before finals, possibly getting ready for standardized tests at the end of the school year, maybe wanting to review science before you go to college in the fall. So many different uses for it, and you get a lot of information, a lot of good materials. All right, thank you. I hope to see you again.